Participants, hope all of you are doing well. So we are going to start in a few minutes. But before that, just let me tell you something, some technical stuff. This class is for hands-on demonstration of bioinformatics databases, and we'll try to demonstrate as many databases as possible, but depending on time. And the next one is I think all of you know that the name of the organization and the name of your classroom is University of Kalyani. So this is the name of the IBT classroom, and it is hosted by me, Angshuman Bagchi, as well as my staff members. I think all of you have the list. And the staff members are Dr. Shima Bishas, Dr. Dipanjan Guho, Mr. Anirban Nondi, Mr. Jeet Mondol, and Mr. Korna Roy. And all of you need to attend the question answer session. And if you have any doubts, you need to put up your questions. So you need to ask your questions in that session. Okay. And if you still have some doubts, you can ask us. And also be sure and make the buttons. It is mandatory that you check the uploaded videos in the Vula platform so that you have a fair amount of idea. So do you have any question? Okay, I think in the chat box. Okay, no. So without further delay, Anirban is going to demonstrate you the databases. Okay, over to you, Anirban. You start the screen sharing and the answer.
Yes, sir. Thank you, sir. Very good morning, everyone. Today, mainly, I will be demonstrating the NCBI database. And if time permits, then I will also demonstrate the BLAST function. Okay. So let's start with the NCBI. Okay. I think my screen is visible.
Yes, it is visible to me. I believe that all the participants can see your screen, right? The participants? The screen is visible to you all?
Yes.
Okay. Thank you. So as you all know, the NCBI, or the National Center for Biotechnology Information, provides us with the access to several databases with biochemical as well as genetic information, and also with some bioinformatics tools. I'll show you how to navigate some of these databases to know more about specific genes and proteins. We'll also take a closer look on how to analyze gene variants as well as gene mutations. So before that, let's start by searching NCBI on your browser. By clicking on the first link, this is the homepage of NCBI. On the left-hand side, as you can see, that we'll find a detailed list of all the resources. See here, of all the resources that we can get access to. And on your very right-hand side, we can get the most popular resources as listed, such as the BLAST tool, which will be showing very shortly.
Why didn't come out Explore classes?
I think someone's mic is on. Can you please kindly mute yourself? Okay, thank you. And in the center of the page, you can find how to submit, or you can download the data. You can also find things to explore, like NCB's research projects and all, like here. And you can also learn that by watching the tutorials. If you are interested in specific genes or proteins, you need to use the search box on top of the page. To the left of the drop-down menu, we have the list of all the databases that are available for research. Depending on what we are searching for, you can choose like genes, proteins, nucleotides, whatever, depending on your research topic. This is the name of if you type that CFTR, that is the gene coding for cystic fibrosis, CFTR. Okay. Now, if we go to the nucleotide, then we'll be searching for only the nucleotide, right? By clicking on the search. the yeah, the results will include all the records that matched your search terms in terms of the selected databases, like the name, from which organism, the accession number, and this is a particular unique identifier of a sequence in a database. And on your left hand side here, you can see you can like filter the results based on your species, molecular types, source databases, and much more, like sequence types, genetic compartments, revision data, and all these stuffs. And on your on your very right hand side, you can search the results by the taxon. See here, you can see the top organisms: Pseudomonas, Homo sapiens, Muscularis, etc. And also by clicking on the advanced button, this is the another way of searching. Here you can refine your results further. If you want to refine your search results, you can enter the organism. For example, if you want to write search for Homo sapiens, let me show you. And all fields type of project biosample, all these stuffs. And another way of searching this, running this type of advanced search, just by going back, and here you can search CF here, then you can type the name of the species. For example, if I type Homo sapiens, it will show, like it will refine the searches. See here, results by taxon, you can see the top organisms is the Homo sapiens, giving results of 1174, right? Now, here we can see all the items that have been listed for Homo sapiens particularly. Items are like 2495, almost 2500. Now you can see that some of the databases contains partial CDS. Now, what is this CDS? CDS is nothing but it is the code for DNA sequencing. Now, but we want a full length, right? So here we can see that partial sequences are present. It will take a bit of time to get for looking through the whole sequences sorry, the results until we get to the right one. Now here you can see the complete sequences. On the 15 or 16, here you can see the complete sequences. And also you can see the length of the DNA is 492, 560, all the SSN number. Now if we click on it, is opening. Yeah. Now it takes us to another database of NCBI called GenBank. See here, you can see that it is GenBank. Now GenBank contains the locus where the chromosome, it is located in this particular gene on the chromosome, the definition, the accession number, when it was published, it was published in 2016, the source organism, the reference authors, the title of this published paper, the journal, the features. Now here the most important features is that you can see the source, the gene, and the gene products or the intron exon portion, and finally the origin of this gene. Now if we click on the FASTA sequence, now why we are clicking on the FASTA sequence? Like while analyzing the protein or nucleotide sequence, they must be in a particular format. And the most common and the oldest format is like the FASTA format. In this format, nucleotides or amino acids are represented by letters. Let's click on the FASTA format, and here you can check. Yeah, here you can check. See that here it is nucleotides. All are represented by letters. Now, one significant features of this FASTA sequence, like how you will identify this is a FASTA. See, it is always headed by a greater than sign. See, greater than, then the accession number, then the species, and then the gene. It is giving us the complete sequence. So either you can copy it and paste it in our respective either Word or Notepad, or you can go to here, send to, file, create file. It will get downloaded. Right. Now we will be going just below that on the now if we want to analyze this sequence, we can write BLAST. We can click on BLAST, and there are several types of BLAST. you can run. Blast N stands for running blast nucleotides. Blast P stands for where we can enter our query protein against a query protein. Blast X stands for like nucleotide translated into protein, and it is being blasted. Now, this blast stands for
Anirban, just
Yes, sir.
one thing, that since there is a network issue, the voice is kind of breaking in between. So can you please repeat the entire thing just in a nutshell?
Yes, sir, sure, sir. So, sir, am I audible now? Clear.
Yes, you are audible now, but the participants are writing that the voice is breaking. Yes, now it's better.
Oh, okay, sir.
Yeah.
Okay, sir. So here we have I have mainly discussed the feature section. After clicking, if we just go back, here we are taking the complete sequence, right? Here we are taking the complete sequence because many are present in partial sequence. So we are interested more in complete sequence. So by clicking on that, it takes us to another web page. Now here we can see it is the another database controlled by NCBI, that is the Gen Bank. Now here you can see the Gen Bank with its accession number. This is a unique identifier. Here you can see the locus, that is on which chromosome, which position the gene is located, the DNA length, and the publication ID or the publication date rather, and the definition, that is the number of species it is there. It is a complete sequence present. And the keywords are here. As you can see, it is a cystic fibrosis transmembrane conductance regulator and the source organism. Now here also we can see find that the authors, the name of the authors are present, the title of this particular research topic. And finally, if we come down, we can see the feature portion where we can see the source organism, the gene, and also the gene products, that is the intron and exons present. And ultimately, here we can see the origin. Right? Here you can see the origin. Now, while analyzing protein or nucleotide sequence, they must be present in a particular format. And the most common and the oldest format is the FASTA format. Like if we come up and click on this link, the FASTA sequence, it will open this type of sequence. And here we can see the every nucleotide or every amino acids are being represented by a single particular letter. And how to identify this is the fastest sequence is that it is being headed by a greater than symbol. See here, you can see the greater than symbol followed by the accession number, the species name, the gene, and ultimately the complete sequence. Then the faster sequence. Now, one way to copy is that we can just copy it and paste it, or we can like save to, and like we can click on send to, we can go to file, and we can just do create file, and it will be copied and pasted. Now, if we search for the protein, like if we go back, here we have searched for nucleotide sequence, right? Here we have done nucleotide sequence. Now, if we want to search for protein, we'll simply go to protein. In the it will be performing the same method. This and again the search button. Here we can see the partial. All we are not interested in the partial, right? So it will take just a bit of time to go through it to find the complete sequence. Now, once the complete sequence is being found, then we can proceed in the same manner as we have proceeded in for the nucleotide sequence. Now, just you can go back, and if we go to that particular sequence, it was like this one. Mm. We want the complete sequence, right? So we can search for that complete sequence. Here it is, or here it is. Yeah, it is loading. Just a bit of time. Yes, here it is. Now, if we on the right hand side, we can see that we have this particular pane, where you can find the analyze this sequence. We can run blast, pick pipe, primers, find in this sequence or highlight some kind of sequence features. Now, in this, like for proteins, in this feature section, if we come down below, here you can see the nucleotide. If we go to for proteins, like proteins, if we search for this particular protein only, EFTR, here also we'll be considering the complete sequence. Mm, yeah, for this sequence, we can see that uh, here we can see the region and the sites for this particular protein. Now here we can also see the sites which are being phosphorylated or the transmembrane regions for the particular protein. Now here we can also find the CDS, or it stands for the coding DNA sequence, which is the portion of that gene that codes for this particular protein. And finally, at the very end, we can see once again the string of letters. This string of letters, we can see it is also similar to the nucleotide sequence, right? Here we can get the FASTA sequence. This is the origin, and this every letter codes for particular amino acids here. Now, if we go back and once again go to the nucleotide thing and give it a search, yeah. So we want to go for CFTR in Homo sapiens. So we search for Homo sapiens. It will give us these results. And we'll once again be taking the complete sequence. Yeah. So now we'll be performing our blast function. Just here you can see that by this analyze this sequence. Here you can see that the find regions of similarity between this sequence and other sequences using blast. So we'll be clicking on it now. Here you can see the types of blast, various types of blast programming present. This blast N, it stands for our query nature. It will be nucleotide, and finally our database against which it is searching for. It will also be kind of nucleotide. Blast P stands for our query nature to be a protein, and it will run a search against another protein. Blast X, this is another very unique thing. This X, Blast X stands for the nucleotide which is being translated into protein, and it will run a search against another protein. T blast N will run a search of a query nature of a protein and will search against a nucleotide. And finally, it comes down to T blast X, which will be searching for nucleotide running against a particular nucleotide translated into protein. Here you can enter our accession number or our FASTA format. Now this accession number corresponds to this one. Here you can see, as I am running BLAST directly from here, so it has taken the accession number directly from here. Or if we don't know the accession number, then we can open our server. We can write ncbi BLAST. Just click on the first link. Yeah, here you can see BLAST X, T BLAST N, protein BLAST, nucleotide BLAST. Here we're running the nucleotide BLAST. See, here you can see the accession number. We can enter our accession number or the FASTA format. Okay, so here it is given already it has taken the accession number. Now, this query subrange means that if we want to give 1 to like 300, if we like 200, if we want to search our query, so it will only run the query for the very first 200 sequences. Here we can also got the option for upload file. We can choose the file for desktop, or you can give the job title is already been taken. Now here is another unique thing. If we want to align 2 or more sequences, which is a multiple sequence alignment, we have to click on this checkbox. See, the interface has changed. Okay, now if we want to deselect it, we can again deselect the checkbox. Yeah. Now, if we want to run this, like if we want to run the similar sequences, MegaBlast is quite a handy tool because it runs the blast for nucleotides. Now, if we want to show the results in new window, we can click on here, algorithm parameters, it is there. All these are experimental specific. For your particular experiments, we can like filter this against species. We can filter them to run your blast, our blast. And all these are experiment specific. And now if we click on this blast, it will run our blast. So you can see the job has been already been submitted. It's searching for the sequences. And ultimately, we'll get the result. Yeah. So this is the result page. It will look like somewhat like this. Now, one of the most important features of this result page is this E value on the this value signifies that the randomness of our particular hits means lower the E value, it will denotes the more number of specific hits it has encountered, right? And the lower the E value, the better it is. You can deselect all. See here we have got the 59 sequences. Our query length is 200. Molecular type is type of nucleic acid, and we have run BLAST N program. So from here we can. So this is the ultimately the BLAST interface which we have done. So over to you, sir, now. So I have got a few questions. It is saying like, oh, query. I have yeah. Query means the portion which we are entering, right? The portion which we are entering is the query, and it will be running against a particular database. Okay. And what is E value is that it is the expected number of, it is particularly a statistical indicator. We can get randomly or by chance alignment here, or we can get the true alignment. Now, if it is kind of true alignment, the value will reduce significantly, which is the best possible result we can expect. And if the value is quite more, like if the E value is more than 10, or then we can see that it is like distant relationship. And if the value comes to like 10 to the power like -20, -50, then we can say that yes, they are like
evolutionary related, if they are got the homologous relationships among them. So that's the significance of the e-value. Basically, it's a statistical parameter. And
Just one thing, that e-value, the lower the e-value, the better is the result, the more reliable is the result. Just this is your take-home message, that whenever you perform a sequence alignment, you need to check the e-value. And if it is very low, your result is reliable. Because see, what you are doing is you are analyzing a sequence, which is just some letters. And no biological information is there. So you have to know whether the result that you are getting is biologically relevant or not. And the e-value is going to give you that. Okay. And any other questions you have? Okay, then one thing that you need to remember is you need to join the question answer session today. The usual time, the time frame is there in the VULA. So you join there and be prepared with your questions. Check the uploaded videos properly. And if you have any question, ask that in the question answer session. Okay. And our next class will be on next Tuesday, the same time, the same Zoom link will be used. Thank you very much for joining. And I think all of you will have a good day. Thank you.
So one thing.
Yes.
I think I want to say something like for the participants only. It would be very encouraged of you all if you can see the tutorials that are provided in the VULA platform, and from now on, for all the Zoom classes that we are having from our end, we will be taking questions from those videos. If you have any doubts or queries, so please let us know about those queries, and we'll be happy to help you for that. As well as if that is all for us, and the question answer sessions is also important to you all, because from there you can also directly interact with the professors who have taken the classes in the VULA platform. So yeah, I wanted to clarify it. Sir has already told you, but from my end I have also informed you guys. Okay.
I think the schedule is already given to all the participants, and if you should activate your VULA platform, VULA account, and it is 4:30 p.m. Indian time, IST, and it is fixed for all the days. Thank you.